Welcome to the 2022 Keys Slam. Interns will present the research they have conducted this summer under the mentorship of their individual labs for 2 minutes each. Keys provides high school students interested in STEM research the opportunity to work in University of Arizona laboratories for 6 weeks during the summer. The initial week of the program is spent training with Keys staff to be ready to begin researching in their lab the second week of the internship. The 55 interns in this 16th Keys cohort were chosen competitively from 294 applicants representing 21 different high schools from across Arizona. The rigorous selection process included a formal review of their writing, teacher recommendations, and an interview with Keys program staff. Getting into Keys is a huge accomplishment. Interns collaborated with scientists, engineering, and physician researchers on research projects as diverse as their interests over the past 7 weeks. We are so proud to share our interns presenting their hard work during these amazing presentations.
Hello, my name is Joseph Cunha-Moreno, and this summer I worked under Dr. Arvinder Sandhu in his Optical Sciences lab. The research question which I explored was polarization-dependent diffraction effects of a reflective grating using a helium neon laser. So first, what does this all mean? Polarization is the electromagnetic field within the laser beam, and a diffraction grating is an optical element that can be used in laser physics to create different diffraction orders in a laser beam. And in my experiment, I focused on the m equals 0, m equals +1, and m equals -1. So to focus on my methods, I control the polarization by using a half-wave plate, and I monitored it by using a high contrast polarizer. From here, I put my data into MATLAB, where I made 3 different graphs. From here, I can analyze my data, where I can make a few conclusions. The first one being is that different diffraction orders can be used to optimize different polarizations. This can be used to guide future attosecond photoabsorption investigations and to study transit absorption and emission spectrum in atoms and molecules. So overall, this is used in optical, nuclear, and atomic sciences, and it's also heavily used in spectroscopy, which is the study of electromagnetic spectrum. Thus, not knowing the response of your diffraction grating to different polarizations can negatively impact the effects onto your spectrum. Thank you.
Morning everyone, my name is Zahraa Alfatlawi, and this summer I interned in Dr. Alicia Allen's lab on how hormones and caregiving behaviors can help women postpartum with a history of substance use. As we know, opioid use disorders are a public health problem, with women in the postpartum period being most vulnerable to relapse. What can we do to help this global issue? Well, the ORCID study focuses on the question, could hormones help women avoid using opioids in the postpartum period? My specific role this summer to help answer this question is working on an expanded literature review on the relationship between oxytocin and opioid use disorder. Oxytocin, also known as the love hormone, has recently been researched about its ability to treat addiction. While we have a focus on OUD, we are also looking at other substances and seeing how oxytocin could potentially affect them. After lots and lots of reading, like 385 out of 6,183 papers, we found that there is still not enough research. A little anticlimactic. However, we did find that the use of intranasal oxytocin actually helped reduce alcohol cravings. This is so important because it leads us to the question, can oxytocin have a similar effect on OUD, opioid use disorder, as it does on alcohol? You may be asking, why is this important? Who is this benefiting? The number of women with OUD at labor and delivery quadrupled from 1999 to 2014 and is only increasing. So many women struggle with substance use disorders, and the postpartum period provides a unique opportunity to help mothers from relapsing. One thing I loved about my lab this summer was how directly impactful it was to so many women. It is vital to destigmatize women with substance use disorders and break down those stereotypes so women, especially marginalized groups, feel comfortable getting the help that they need and deserve. Thank you.
Hello, my name is Gregory Allen, and over the summer I interned for Dr. Nima Tusizadeh. His lab discusses and investigates physiological indicators for frailty, which is defined as a clinical syndrome in which 3 or more of the following criteria are met: unintentional weight loss of 10 pounds or more in the last year, weakness, slowness, self-reported exhaustion, and low physical activity. My research specifically focused on calculating heart rate dynamics for the control subjects and patients' intervals of rest and movement through the electrocardiogram data in the HRV program, and calculating Shannon and sample entropy, which are 2 different ways of calculating heart rate complexity, and it is closely associated with frailty. The control subjects were healthy, non-frail young individuals, and the patients were older individuals with congestive heart failure, a decline in the normal pumping power of the heart, which is closely associated with frailty. Our results show that, on average, the control subjects had a higher sample entropy in both intervals, and that these results were statistically significant. On the other hand, there was no statistical difference between the Shannon entropies for both the controls and subjects for both intervals, for the controls and patients for both intervals. This means that heart rate complexity, measured by sample entropy, may possibly be used as a physiological indicator for frailty. Our lab's research is very important because some medical treatments and therapies are not safe for individuals who are frail, and predicting frailty within older individuals can help prevent those people from getting those treatments and therapies. Thank you.
Hi, my name is Vishruth Anand, and this summer I worked in Dr. Etkin's Memory Development and Disorders Lab, which focuses on atypical development, specifically Down syndrome, which is the most common genetic cause of intellectual disability and can lead to memory impairment and other developmental issues. Autopsies of people with Down syndrome show a smaller hippocampus, which is the brain center for learning and memory, and studies that attempt to analyze this throughout development are inconclusive, and we think it's because they are not looking at the subfields within the hippocampus. In order to compare the relative development of the subfields between typically developing people and people with Down syndrome, we took MRIs of participants ages 11 through 20, and then we try to estimate the volume of the subfields. We use ITK Snap, which is an imaging software, along with anatomical markers, measurements, and Dr. Arne Ekstrom's protocol in order to estimate the volume of the subfields. We had multiple raters doing this. In order to ensure inter-rater reliability, we did a test which had results over 95%. We're looking at the CA and the DG regions, which are 2 subfields that are extremely important in memory. We found that, our hypothesis was that our hippocampus volume in the CA and DG would be lower in Down syndrome. And we found that in our CA anterior region, there was no statistical significance, but there was a higher variance in typically developing people. In our CA1 and our CA3 posterior regions, however, our hypothesis was proven right, and our Down syndrome group had a smaller volume. This is really important, and it's a pilot study, so as we do more, we'll have more information on it. But we can use this in future neuroscience research, psychology research, and other data. So we have cognitive tests that we're running on the side, and we're going to be using these cognitive tests to see strengths and weaknesses in cognition compared with our brain data. Then we're also taking our sleep data using an ActiWatch, and we're looking at how there's sleep disruptions in people with Down syndrome. Finally, we're going to compare sex differences, and we're also going to do functional connectivity data, which shows which parts of the brain have activation at the same time of these subfields. So what we've done right now is just the start of this project, and we can use this in education and future neuroscience researchers to learn so much more about this disease. Thank you.
Hello everyone, my name is Pranav Barla, and this summer I worked under the guidance of Brenna Reinheimer and Kirsten Limesand. Thousands of people suffer from the loss of salivary gland function after undergoing radiation treatment for head and neck cancer. Radiation treatment in these patients causes decreased salivary function and in turn causes many oral problems such as tooth decay, gum swelling, trouble swallowing, etc. The Limesand lab studies ways to improve these patients' quality of life. And in this study, we looked at proliferation or cell division of the salivary gland cells in mice to better understand the wound healing process of the gland. We looked at previous research of proliferation suggests that mice cells start proliferating 5 days after radiation and continue at this elevated level through chronic time points. We studied the effects of YAP, and we used Ki67 as a marker of proliferation. YAP activates the transcription factors of genes and is necessary for wound healing in other tissues, and is also at its highest level in glands that do not regenerate. We also looked at EDU-positive label-retaining cells, which are indicative of the heterogeneous population of salivary progenitor cells. Our overall goal was to find the balance of YAP needed for maximum proliferation, and we asked the question, how is proliferation affected by the presence and absence of YAP after irradiation of the head and neck regions in mice? 4 to 6 week old DCPP1 Cree YAP mice were treated with 5 grays of radiation, and salivary glands were taken out for histology. The percent dual positive Ki67 EDU over total positive EDU does not differ with the presence or absence of YAP. We also looked at the effect of verteporfin on the salivary flow rate of mice. Verteporfin is a YAP inhibitor, and we give 3 doses 5 to 7 days after irradiation. Previous research on verteporfin suggests that that salivary flow rate decreases after irradiation, and verteporfin has no impact on untreated salivary glands. However, verteporfin does have a significant impact after 30 days of irradiation and leads to an increased salivary flow rate, similar to that of untreated glands. Therefore, we can conclude that the inhibition of YAP restores salivary flow rate similar to untreated glands, and this makes verteporfin a very viable treatment for cancer patients, allowing for better quality of life. Thank you.
Hi, my name is Malcolm Barrios, and this summer I worked in the Meredith Lab, which focuses on environmental science research. So my project asked the question, does water harvesting practice affect soil moisture resiliency? In this case, soil moisture resiliency refers to how the soil returns to an equilibrium of moisture level after a pulse event. So here in the desert Southwest, water is a really limited resource, and therefore any water event such as irrigation or rainfall results in a spike in moisture and then a slow return to equilibrium. So 3 treatments of water harvesting were tested. We tested a control, which was unaltered soil, a passive treatment, which was a water collection basin, and then a gray water treatment, which was a collection basin that was also irrigated by washing machine gray water. So, a paper, Todman et al. 2016, was identified using pulse dynamics and resilience theory, and it calculated 4 main variables of resilience. So those four dimensions are return time, the time it takes for the system to return to equilibrium, the degree of return, if it returns to its original baseline or a different baseline, the average slope, the rate at which it returns, and then the area under the curve that is modeled. So I calculated these 4 dimensions for my 3 treatments, and then my results were produced. So I found that my hypothesis was supported. The gray water treatment had the most efficient response with the least area under the curve. Other important findings were that the passive treatment had the least efficient response, and it took the most time to return to baseline, meaning the moisture lingered in the system. That means that there could be more moisture available for a longer time for plants and animals in the ecosystem, which would be beneficial. Or pulse events are known to release CO2 into the atmosphere, so lingering moisture level could respond with lingering CO2 release, which wouldn't be beneficial. Thank you.
Hello everyone, I'm Vinyas, and this summer I worked with Dr. Jennifer Barton. The Barton Lab develops falloposcopes, which are endoscopes to enter the fallopian tubes to image for early stage ovarian cancer before it's even spread to the ovaries, at which point the patient outcomes are a lot better. The problem is the fallopian tubes are filled with these small finger-like projections called plicae, which can obstruct or entangle with the endoscope, and so they need to be moved out of the way. One solution that's commonly used with other endoscopies is the saline flush, but it hasn't yet been tested for use in falloposcopy. So this summer I built a flow chamber, which I then used to run saline of varying flow rates over samples of fallopian tissue from pigs. We observed plicae movement under flush with an OCT system, which stands for optical coherence tomography. It's a type of small-scale, high-resolution 3D imaging system. With the OCT, we took videos of the plicae moving under flush. And from those videos, we took specific frames from before and during the flush. In each image, we measured the area of the that the plique occupied, and found displacement by taking the difference between the areas from before and during the flush for each flow rate. What we found was a generally positive trend of increasing displacement with increasing flow rate, but that displacement was small and generally unpredictable. So we conclude that saline flush alone will likely not produce sufficient plique displacement for use in phalloscopy. We did notice, though, certain motions such as loosening and unfolding of the plique, which could be useful for imaging areas at the bases of the plique that might otherwise be obscured. Finally, what this means for the future is that other alternative methods of plique displacement, such as an inverting balloon, should be further investigated in order to ensure phalloscopy success.
Hi everyone, my name is Daveena Biswas, and this summer I worked in the MARDI lab under Dr. Henry Sanders, and we aim to look at how alpha-synuclein mutated proteins interact with the lipid bilayer and how EGCG can affect this interaction. So alpha-synuclein is a protein found abundant in the brain with implications in Parkinson's disease, and when this protein interacts with the lipid bilayer, it causes a conformational change in the shape of the protein, leading to aggregation and toxicity, making this a very important but difficult protein to study. So in the past weeks, we were able to successfully grow and extract our proteins, as well as begin purification of our proteins. And in the future, we aim to expose our proteins to nanodiscs, which are a synthetic model used to mimic the structure of the lipid bilayer. And then we will expose our protein and nanodisc to the mass spectrometer to analyze the interactions between the protein and nanodisc, as well as the interactions between the protein nanodisc complex and EGCG. So when we exposed our proteins to the ion exchange column and size exclusion column, we received high absorbance values, thus indicating that our protein had come out of the instrument successfully. However, when we put our sample through the mass spectrometer, we received abnormally high mass values, thus indicating that our protein was still contaminated or had already aggregated. So this is still something we're currently looking into. And by the end of the project, we hope to see that the proteins bind to the nanodiscs as we have seen in the past with the wild type, and that EGCG inhibits the disruption of the nanodisc from the effects of the alpha-synuclein mutated protein. So I would like to thank the MARDI Lab for hosting me this summer, as well as Keys and McNuggies for being so supportive throughout this entire process. Thank you.
Good morning, I'm Savannah Botello, and I interned at the Meredith Lab of Ecosystem Genomics, where I studied sphagnum moss. Although a seemingly simple organism, moss can contribute up to 50% of total ecosystem photosynthesis, or GPP, gross primary productivity, and yet is excluded from all land surface models, the same models that are supposed to predict things like GPP. So to fix this issue, our study aims to characterize moss gas exchange by 1, modeling the moss drying rate, and 2, measuring carbonyl sulfide gas, or OCS flux, as moss dries in order to really understand if moisture is a control of moss photosynthesis or not. And since we're focusing on gas exchange, you might think that we should track CO2, but photosynthesis and respiration are co-occurring, so it's really difficult to distinguish between both processes. So we use OCS as a tracer gas because it follows the same pathway into the plant, it's naturally occurring, and doesn't have any way out. So for the drying experiment, we wet and dried both living and dead moss. And the results show that the models have a high linear fit, though the dried moss does have a better fit. It has a faster rate of drying based off the slope, and it loses more water than the living moss. We can attribute the fact that the living moss had a higher water retention because it was still alive. And then for the flux experiment, for context, a rising OCS flux is emission and a lower one is uptake. So in this graph, we can see that there was an initial unexpected emission over time elapsed from the moss. The OCS flux also has a negative linear relationship with time elapsed and a positive linear relationship with moisture. Although we can't make any solid conclusions or explanations because these were unexpected results, we can say that moss has moisture as a control of its photosynthesis because of the differences between the wet and dried. Thank you.
Hi, my name is Wesly Carbonneau, and this summer I worked in Dr. Craig Aspinwall's biochemistry lab, working on using scanning ion conductance spectroscopy to image human platelets. Scanning ion conductance spectroscopy, or SICM, is a way to image live human platelets without interfering with the process of activation. Human platelets are large cells in your blood that, when activated by pressure or other chemical sensors, reach out to other platelets to grab on and to form a connection. This connection causes things like scabs to slow or stop bleeding. The problem with platelets, though, when they activate, is that sometimes they'll activate in your blood vessels or in your brain. And this can cause severe damage, usually in the form of strokes. So SICM is a method that involves two electrodes, one inside a bath with the platelet sample, and another one inside of the pipette, of a glass pipette with a tip of about 100 microns. When the pipette gets extremely close to the sample, it cuts off that current. And we can use that cutoff in current to create a three-dimensional image of extremely small objects without touching them, such as platelets. These SICM gives us these measurements that we can then use to study physical changes of platelets over time. My lab specifically used osmotic stress experiments that involved changing the makeup of the bath sample around it that actually slowed the activation of platelets over time, and we're able to look at that through SICM without causing any pressure in activating them. SICM, the measurements that we use are area, so a two-dimensional mass intake, and then we use volume, which includes the height data. Then using those two, we can find circularity and hemisphericity, which tell us how activated the platelets are, because they remain in a general circular shape before activation and over time lose that circular shape. This research and data can be used in further studies with platelets to possibly help create different medical treatments for things such as blood clots and strokes. Thank you.
Hello everyone, my name is Yash Chatha, and I've been researching cognitive aging in Dr. Bonnie LaFleur's biostatistics lab over the summer. One of the most impactful issues with our aging process is that our physical bodies are able to survive for a long time due to the advancements in medicine, but our cognitive health ends up lagging behind. And a recent initiative created to research this cognitive aging was the Precision Aging Network, or we can call this PAN. And this aims to assess our normal cognitive aging process and attempts to provide specific treatments for the different risk categories of cognitive aging. To build a better foundation for this PAN study, we compared datasets from the Alzheimer's Disease Neuroimaging Initiative, which we can call ADNI, and the National Alzheimer's Coordinating Center, or the NACC. And these datasets include information on patient demographics and cognitive aging stage. We also evaluated the differences between concentrations of 3 biomarkers which can indicate cognitive aging progression. Using R, we did an analysis between these two datasets, and then we generated visualizations to work towards a better understanding of these patient profiles in each dataset. One of the trends we noticed similar across both datasets was that patients later on within the normal cognitive aging process tended to be male, while patients with relatively normal cognition tended to be female, and this could indicate an error in patient recruitment. Another trend was that NACC dropout rates were typically higher earlier within the study, while ADNI dropout rates were typically higher later within the study. In addition to this, I also evaluated the longitudinal changes of these biomarker concentrations, which are alpha beta, phosphorylated tau, and total tau, using our ADNI data within this 5-year period. And we found that the most significant differences in baseline concentrations between these diagnosis groups occurred with the biomarker phosphorylated tau. All 3 biomarkers are not statistically different in concentration by the end of this 5-year period, and this is likely due to the increased margins of error due to a smaller sample size over time. By the end of the study, I was able to harmonize both the ADNI and NACC datasets by this diagnosis type, and I was able to evaluate the differences in trends between these 3 biomarkers. This study that we just worked on can provide a solid basis for the Precision Aging Network and the patient recruitment process, and it can provide validation benchmarks for future modeling of the Precision Aging Network data. Thank you for your time.
So, picture this. A woman who is suffering from ovarian hormones to experience the worst pain she's ever experienced in her whole life. She might be tempted to engage in opioid use to stop the pain. Anything to make it go away, right? Well, the ORCID study is here to help her. My name is Riana Chatterjee, and this summer I interned in Dr. Alicia Allen's ORCID study with the Renew team. The goal of the ORCID study was to analyze the role that caregiving mechanisms have on hormonal alterations, which could potentially lead to cessation from opioid use disorder. We are essentially answering the question of how caregiving mechanisms like breastfeeding and skin-to-skin contact can influence the hormone prolactin, which would lead to this cessation. My specific role in the project was the review of literature. Specifically, I analyzed 150 research papers to determine the relationship between breastfeeding and prolactin. My part analyzed 50 papers. Through various rounds of research and elimination, I ended up with 15 specific articles that mention a relationship between these two variables. The reason that the hormone prolactin was significant to our study was because it showed an exaggerated care response in the maternal brain. What this means is that an increase in prolactin led to an increase in nest-building activity in animals and lactation through milk production in humans, as well as maternal motivation and aggression. This not only protected the infant from predation, but also led to improved mother-infant health. The next step of the ORCID study is to actually execute this project in our lab and engage in weekly meetings with our mothers to see if an increase in breastfeeding did lead to an increase in prolactin and if this overall alteration is helping them withdraw from opioid use addiction. Until then, however, our research is a significant step towards improving the lives of millions of women all over the world who are looking for alternatives to substance use disorder. Thank you.
Hi everyone, my name is Abhinav Chede, and for the past summer, I worked under Dr. Sawyer's and Thomas Knapp's guidance in a biomedical optics and optical measurement laboratory, where they explore how optical tools can be used to improve disease diagnosis. In this internship, I worked on duodenal gastrinomas, which are a type of tumor that originates in the small intestine. It's thought that these tumors come from abnormalities in Brunner's glands, which are small glands in the small intestine. Being this small, it is hard to accurately image these glands without altering the sample. Next slide, please. Here enters a promising method called multiphoton microscopy, or MPM. MPM is an imaging tool that helps the user accurately observe texture features of a tissue without invasive procedures or reactive contrast agents. I specifically investigated if there was room for the use of machine learning to automate and improve MPM diagnosis of these tumors. My mentor collected the MPM tissue samples, and I quantified certain image features with the software called ImageJ. Next slide, please. Using Python, I ran statistical tests on the data and generated a machine learning model that uses the quantified data as inputs to tell if the tissue is cancerous or not. I then created an image classifier model that takes the collected MPM images as inputs and similarly decides if the gland is abnormal or not. Both these models had accuracies in the high 80s, and seeing this high accuracy, it can be reasonably deduced that there is an application of machine learning to improve gastrinoma detection in a clinical setting. Thank you.
Good morning, I'm Carol, and this summer I was in the Carnes lab under Dr. Jason Carnes working on warfarin pharmacogenomics, or the study of how your genetics can affect your drug response. So warfarin is a common anticoagulant or blood thinner used to treat patients with blood clotting disorders. Proper dosing is important because too much can cause bleeding, while too little doesn't properly address the blood clotting issue. Both of them can be fatal. Warfarin in particular is a difficult drug to get accurate dosing for because the dosage varies widely from patient to patient, and the drug has a very narrow effective beneficial window. So there are identified genetic variants in the warfarin pathway that would affect warfarin dosing. For example, the CYP2C9 gene is coding for the enzyme that metabolizes warfarin, and the STAR3 allele it reduces the enzyme activity, which causes warfarin to be cleared out slower and patients requiring less warfarin. So genotype-guided dosing algorithms consider clinical factors and these genetic factors to calculate more accurate dosing for patients. However, fixed dosing, or giving each patient the same amount of warfarin, is still the clinical standard, even though it is less accurate. So we genotyped a previously identified genetic variants that affect warfarin dose, and we replicated the previous genotyping results. We also looked at genetic variants and their effects on warfarin dosing, and we found that the CYP2C9 STAR3 allele is associated with a lower weekly warfarin dose. So if you look on the graph, you can see that patients without the risk allele have a higher median than patients who are homozygous for the risk allele. So in conclusion, this validation of these variants in a diverse study population strengthens evidence for clinical implementation of genotype-guided dosing algorithms.
Hi, I'm Raiyan Choudhury, and this summer I'm working with Dr. Vinyas Bhat at the Computational Medicine and Informatics Co-Laboratory, who's specifically my project revolves around long COVID. As we all know, the COVID-19 pandemic has caused a global healthcare crisis with over 6 million fatalities. For those who survive, the acute COVID-19 has led to the persistence of symptoms for 4 weeks after the initial infection in up to 30% of patients. This condition, known as long COVID or scientifically as post-acute sequelae of SARS-CoV-2 infection or PASC, has manifested symptoms across multiple organ systems, significantly disrupting the lives of affected patients, making it a critical healthcare concern. Unfortunately, there are currently limited to no characteristics that can holistically categorize all aspects of PASC. Thus, my project aims to help this disparity by creating an ontology. An ontology is essentially a formal model to help visualize relationships and data. By doing so, we can help better understand PASC to help eventually solve for it. To create my ontology, I first did extensive background research on the underlying factors and symptoms of PASC to better understand it. I then began designing my ontology by creating various modules and axioms of Projet, which serve as the kind of classes and aspects of PASC, such as the symptoms experience, the underlying factors, and so forth. And from there, you can kind of visualize the relationships between them. After creating this ontology, I then validated its efficacy by implementing sample patient data into the ontology and categorizing it. Moving forward, my ontology can serve as a critical first step in helping research to better understand PASC and help solve for it. This is because ontologies are really good for visualizing data. So by using this ontology, so this one, researchers can help develop the most prevalent characteristics and trends in PASC, which can then guide further solutions, ultimately helping millions of people. Thank you.
Hi everyone, my name is Erin Conant, and this summer I interned with Dr. Jennifer Teskey. So obesity is extremely prevalent in the United States, and it is a co-occurring epidemic with type 2 diabetes. Now the American diet has high-fat foods. Often it also has high glycemic index foods, such as potatoes. To evaluate this issue, my lab is experimenting with a low glycemic potato to see its effect on weight gain in a rodent model. Now to do this, we have 80 rats, 40 males and 40 females, and these rats were divided into a high-fat or low-fat diet. These diets were further subdivided into high or low glycemic index potato diets in addition to the first diet. Now, after all 5 weeks were complete, the rats were measured for blood glucose levels and insulin levels. Now, in our results, in the first half of the results, we can see that the high-fat individuals in males and the low-fat individuals in males showed a difference. However, our females did not show a difference in fat groups, which was slightly unexpected. This is also reflected in our calorie intake and in our percent body fat composition. In the second half of the study, figure 2, we see that our results are very varied, despite expecting that high glycemic index potatoes would cause a higher weight gain in all individuals on them. This is obviously not displayed, and you can see that the low-fat males had the exact opposite, in which the high-fat potatoes had a lower weight gain than the high glycemic potatoes had a lower weight gain than the low glycemic potatoes. Now, we have an explanation for this because these results are extremely preliminary. They occurred on day 5 out of 14 days of the second half of the study, so they're fairly inconclusive in comparison to our final results. Now, these results, when completed, will indicate whether or not we should perform a human trial, and maybe even in the future incorporate low glycemic foods into the American diet, lowering rates of obesity and type 2 diabetes across our nation. Thank you.
Hi, my name is Momoko Cronin, and this summer I interned with the Giannetti Lab, and my project focuses on creating tethered dual photocatalysts. What are those? Photocatalysts are a substance that, when excited by light, affects the rate of a reaction or the feasibility of a reaction. And what that means is that it's going to speed up a reaction or make that reaction possible in the first place. Dual means that it has an organic and metal component, and tethered means that those components are physically linked to one another. The reason why we want to create tethered dual photocatalysts is because it's going to be more eco-friendly and cost-effective than organic and metal catalysts alone. We hypothesized that in order to create our tethered dual photocatalyst, we would have our organic catalyst, and we attach ligands to it, which is kind of like an arm, and we attach ligands in different places and make them longer, and then when we're finally satisfied with what we have, we would eventually attach a metal to the complex. So our results showed that our attempts at attaching ligands to an organic catalyst were successful. We've made a number of different organic catalysts with a number of different ligands. In my presentation, we have 4 examples of what we've successfully created thus far. As for organic catalysts and our metal catalyst binding, we've confirmed that we're able to bind one of our organic catalysts to nickel. However, we're not sure if that binding resulted in the structure that we want, so we need to do a larger scale reaction and X-ray crystallography in order to confirm a successful tethered dual photocatalyst. Thank you!
Hello. I'm Tej Desai, and this past summer, I worked in Dr. Bonnie LaFleur's biostatistics lab, where we did a crosswalk evaluation between participant data from 2 longitudinal studies from the National Alzheimer's Coordinating Center, or NACC, and the Alzheimer's Disease Neuroimaging Initiative, or ADNI. As humans age, our cognitive function levels change or differ, creating a gap between cognitive health and chronological aging. The University of Arizona's Precision Aging Network, or PAN, actually works towards and better understanding cognitive health through their own longitudinal study of participants, which is exactly where our lab and project come into play. We looked at NACC and ADNI data, both of which required the use of highly complex statistical analysis, and we split the project up into 2 main objectives. The first was a data harmonization process, where we had to sort of align the 2 data sets based on the common diagnoses of normal cognition, mild cognitive impairment, and dementia, as well as looked at the patient dropout rate from baseline, which is the initial visit, and patient diagnosis transition from baseline to year 1. For the second objective, we looked to see if there was any association between cognitive aging and comorbidities such as diabetes, stroke, and even hypertension. Now, the results of the study were quite interesting. We saw that with the NACC dataset, there was a higher percentage of participants dropping out earlier in the study, in like the first few years. We saw that there was more participants actually transitioning their diagnoses from cognitively normal to mild cognitive impairment and dementia. And for the second objective, we used multiple logistic regression analyses to generate plots and concluded that hypertension and stroke were the 2 most closely associated comorbidities with cognitive aging. So overall, these results will benefit the Precision Aging Network, as the findings from our comorbidity association and data harmonization will be used as a sort of benchmark for future validation for the program's statistical modeling process. Thank you.
Hi, my name is Jack Douglas, and over the summer, I worked in Dr. Dornhaus's social insect lab. And if you join me for a moment, I'd like to go on a brief mental field trip. So imagine that you and 20 of your best friends are tasked with searching a very large grassy area for a few gold coins. But the catch is you can neither communicate nor see your friends during the search process. What steps should you take to make this more efficient? And that's precisely the question that ants have to answer every day in their daily struggle for survival. And when talking about ant search behavior, it's really important to know that ants passively emit pheromones when they walk, and this means their nestmates can see where they've been, and in terms of searching, this means that they can see past expeditions. And our cited literature says that this is very influential in the search behavior of ants, because if you know your friend has gone down this path and didn't find anything, you wouldn't go down the same path. And so we wanted to see if this trend held true for Temnothorax regatulus, a species of rock ant native to the western United States. So in pursuit of this, we tracked a colony of T. regatulus for 2 separate 5-hour periods. In trial 1, they foraged in an arena that was populated with these pheromone trails, and in trial 2, they foraged in a blank arena. And we actually found some curious results. First off, there was no significant difference in tortuosity between trials, and this suggests to us that the ants did not, in fact, use the existing pheromone trails to expedite their search process. However, there was a very large difference in terms of the average speed between trials, and it was in fact trial 2 ants that moved faster, opposite to our hypothesis. We came up with two possible reasons for why this might be. The first one is that the lack of pheromone trails in trial 2 may have essentially coerced the ants into saying the land is contested and unknown. And since competition is such a large part of ant species, or ant ecology, I should say, this may have led the ants to search faster in an effort to claim this land for the colony. And for the second reason, you must know that Trial 2 took place chronologically after Trial 1. And this means that the ants were more familiar with the arena by the time the second trial came around. And perhaps it was this difference in familiarity that accounts for the difference in speed, not the presence or lack thereof of pheromone trails. And if we wanted to investigate any of these hypotheses, we would have to take a more nuanced and microscopic look at how search behavior changes for these ants when they are actively moving on versus moving off of pheromone trails. Thank you.
Hi everyone, my name is Hannah Feinberg and I worked in Dr. Robin Polt's lab this summer. So I think we're all aware of the opioid crisis and how it continues to become an even more pressing issue as overdose deaths increase each year according to the CDC. Glycopeptide drugs offer a super cool alternative to conventional pain medications. So these are strings of amino acids, which are subunits of proteins, with a sugar attached to the end. And instead of breaking down into toxic substances like regular pharmaceuticals would, they break down into amino acids, and these are found all over the body. I helped guide future drug design of my lab's lead compound, SS-oxy-6, by creating different versions of the molecule and measuring the energy with which it binds to the receptor. So SS-oxy-6 is inspired by the hormone oxytocin, has many characteristics that we probably know a little bit about, but the property that we focused on was its pain-killing property. However, the only issue is oxytocin has a very short half-life. What my lab did to fix this is they replaced the weak disulfide bond with a strong, robust amide bond, and they added a sugar to the end of the molecule. And this changed the half-life from about 8 minutes to multiple hours. So what did I do? I used the software system Mo to create 19 3D analogs of the SS-oxy-6 molecule by changing the tyrosine, one of the 10 amino acids that made up the molecule. I then measured the energy of the molecule itself, and then the molecule bound to the receptor, and we'll just call that the complex. When we measure total energy, we want a lower total energy, because that means less energy is needed to keep the system in place and the interactions more likely to occur. What we found was that low energy molecules didn't necessarily have low energy complexes, and ultimately that's what's most important, because how a molecule binds to its receptor is going to drastically change the effects it's able to create. So all in all, I did a small part in the grand scheme of my lab, but small changes to molecules can help us create even better drugs to help us fight the opioid crisis seen in our country and around the world. Thank you.
Hello everyone, my name is Ben Gerber, and this summer I worked in the biochemistry lab of Dr. Michael F. Brown. We studied the G-protein coupled receptor, or GPCR for short, rhodopsin. GPCRs are important because when they malfunction in our body, things go wrong and we end up with diseases like diabetes and a couple of types of cancer. For this reason, GPCRs are targeted by 34% of all FDA-approved pharmaceuticals. Rhodopsin itself is important because it's the protein in our eyes that allows us to see in dim light environments. Rhodopsin is activated when light or photons come into contact with the protein and are absorbed by its retinylidene ligand chromophore, which is just a fancy way of saying the part of the protein that triggers the activation. After this activation, a broader conformational change in the protein is encouraged. My goal for the summer was to understand this activation and the changes that follow. The Brown Lab knows a little bit about this already. They've determined that upon activation, an influx of 80 water molecules occurs and rushes to the core of rhodopsin. We approach this investigation from a computational perspective. We used molecular dynamics trajectories from our collaborators and simulated a membrane protein system surrounded by water. We approached this investigation from a computational perspective. We modeled molecular dynamics trajectories from our collaborators and simulated a protein membrane system surrounded by water. We modeled this system over 4,000 steps, or 4 microseconds, and then counted the water molecules inside of the protein in its inactive and active states. Contrary to experimental results, we were unable to determine any significant difference in water molecules between the inactive and active states. We also modeled the electronics of the rhodopsin active site. We selected specific amino acid residues from the protein that have direct influence on the retinylidene ligand chromophore and its electronics. We simulated this small model system in the Gaussian computer program and were able to produce qualitatively expected predictions of the retinylidene ligand chromophore and its electronics. However, due to time constraints, our quantitative calculations are still running. Understanding these two aspects of rhodopsin activation will lead to greater rhodopsin modeling in the future, and a greater understanding of G protein coupled receptors as a whole. Thank you all.
Hi, my name is Joseph Hahn, and to start off, I'm going to go out on a limb and say you probably had a bacterial infection at some point in your life, whether it was strep throat or even food poisoning. Now, the most common response to any of these issues is to apply antibiotics, such as Neosporin. You apply it, and then within a week or so, you're healed. While this is extremely beneficial for you, it does have an unintended consequence: antibiotic resistance. Now, antibiotic resistance occurs when an antibiotic with a wide spectrum of target bacterial cells is used so frequently that the bacteria it's fighting against becomes resistant to it, hence the name. And this has been an ongoing problem for decades at this point, and this is where the Balchus lab steps in. This summer, I worked in Dr. Balchus' lab, where he studies bacterial interactions with other organisms such as fungi. More specifically, I study talisins, a unique type of bacteriocin which is able to kill bacteria in a more effective manner by simply landing on the membrane and not having to actually infiltrate the cell and stop its internal processes like other antibiotics would. And so, my project focused on trying to genetically prove talisin-based killings in E. coli O157H7, a bacteria strand most commonly known for its role in food poisoning. And so to accomplish this, we tried to mutate a talisin-producing bacteria cell and test to see whether it maintained its killing capacities. Now, we actually didn't use E. coli O157H7 just because it is a little more dangerous to work with, so we actually used DBO5 and 330 in hopes that if the mutation appears within these strands, it will also appear in the E. coli. So, unfortunately, we were unable to effectively mutate the bacteria cell. However, we were able to test a variety of different factors, such as streptomycin levels and induction. And so just to elaborate on one of the other tests we did, we actually tested a mutation method. And so we used conjugation, and we found out that it's more effective than electroporation. And just to expand on that, conjugation is where we mix bacteria together, and electroporation is where we use electricity to poke holes in the cell and have them combine DNA that way. And so further research into the field and talisins specifically would be extremely beneficial, as it could be a potential solution to the rising problem of antibiotic resistance. Thank you.
Hello, my name is Michelle Hine. Over the summer, I've been working in Dr. Melanie Culver's Conservation Genetics Lab under the mentorship of Natalie Payne and Dr. Carla Vargas. I've been researching the relationship among crested caracaras, which are a type of raptor living in South and Central America, as well as Arizona, Texas, and Florida. These birds are one of 1,300 on the U.S. Fish and Wildlife Service's list of threatened or endangered groups, making them of great interest to conservation biologists. But while previous research has studied the effects of habitat degradation on dispersal and mating, this study, our study, is the first to focus on the genetic relatedness of crested caracara populations in the United States. Thus, the ultimate goal of this project was to guide conservation efforts and management decisions. I used related in our package to estimate the relatedness across populations. I found that Arizona and Texas could be somewhat related, but this is unlikely. When individually assessed, I found that Texas had no relatedness, and Arizona demonstrated some subship. What I think is more interesting, though, is that my estimates support that the Florida population is isolated, and this isolation, this genetic distinctiveness, is incredibly important to consider when creating effective conservation plans. I also used sequoia and neck draw to reconstruct pedigrees. Pedigree reconstruction revealed that caracaras are in fact monogamous. This aligns with previous observations. However, sequoia was not able to pick up on at least one known relationship in Arizona and one in Florida. Further filtering, though, could help resolve this issue and create a more robust pedigree. Ultimately, understanding the sexual relationships of caracaras and Florida's isolation from other populations is above all significant in creating an effective management plan and preserving biodiversity. Thank you.
My name is Andrea Hernandez, and this summer I work in Dr. Tomasiak's lab, which studies the metabolism and cancer in membrane transporters in drug resistance. High temperatures promote the spread of diseases, such as Pali fever, a fungal respiratory infection for the southwestern USA, Central, and South America. When the fungus is inhaled, it remains in the lungs in the form of a spherule, a thick cell wall that the fungus uses as a shield, making it difficult to eradicate. Scientists have identified that the protein called carbonyl phosphate synthetase 1, better known as CPS1, is a crucial factor for valley fever virulence. However, its functioning was unknown until my laboratory decided to get down to work. During my project, we studied the structure and mechanism of CPS1, and by using thermoshift ices in purified CPS1, we demonstrated that ATP binds and stabilizes CPS1 by a 2-degree change in thermostability. Also, we compared the activeness of CPS1 by measuring how much ATP consumption occurs in the presence of palmitic acid and CoA, which are suspected to interact with CPS1. Our results show a higher activity when CPS1 correlates with ATP, a fatty acid, and CoA, indicating the possible formation of acyl-CoA, which is one of the building blocks of the cell wall. Our work will contribute to the implementation of prevention measurements against valley fever and would aid in the creation of a vaccine that improves the quality of life and safety of millions of people across the continent of America. Thank you.
Hi, my name is Raiyan Islam, and I've been working in the Biosensors Lab under Dr. Yoon and Katie, located over at Marley Building this summer. These past 7 weeks, I've been working towards refining a device that can detect skin diseases before symptoms even occur, and does this by using a smartphone. The way this works is by using autofluorescence, which is when you shine light at bacteria and it returns a certain wavelength or hue of light back, which can be analyzed for later. In our experiments, we used 2 types of Staphylococcus bacteria, one of them being cons, which represents a healthy sample, and the other being S. aureus, which represents an infected sample with a skin disease. These bacteria were analyzed and imaged under various color sliders using the device, and this was to isolate the wavelengths that came back from the autofluorescence. Then we took these images and put it into ImageJ analysis software to isolate the hue values. In our results, we returned 2 significant trends that can be viewed on the charts on the slides. The blue bar represents the healthy sample, and you can see that it is actually much higher than the other 2, which represents an infected sample. This means the hue value returned was different. In the future, a procedure can be created in which a skin swab sample is taken, processed, and analyzed all in under 15 minutes. And in the future, this device will be refined for optimized user-friendliness, and image analysis software will be replaced by a CNN algorithm for better identification of trends. Thank you.
Hello, my name is Alexander Jauregui-Galarza, and this summer I worked in the Chem lab. Have you ever wondered if there is a way to control cells, direct their behavior, and even a way to mimic living tissue? Well, hydrogels are the catalyst to achieve all these biomedical goals and more. Simply put, the future. What hydrogels are these 3D networks of polymers that are all cross-linked together, and alongside other additives, are suspended in water to form a solid. Our lab has previously found that a gel's mechanical strength can be targeted to natural tissues, which is why they can be used as artificial tissues in the first place. So the first question our lab had was, does protein sequence impact this mechanical strength? In order to test and control this, we use elastin-like polypeptides. Shortened as ELPs, they are artificial polymers that mimic the properties of natural elastin in your skin or any part that requires elasticity. A defining feature of these ELPs are their thermoresponsiveness, where under their LCST temperature they are soluble, and above that temperature they are insoluble and begin to aggregate. So the next question we have is, how does amino sequence impact this thermoresponsiveness? To test this, we use UV-vis spectrometry to beam light into our samples and find the mixture's LCST temperature. Results show that higher concentrations had a lower LCST and more aggregation. We were also able to find linear relationships between LCST and the ELP's concentration, impacting how proteins behave at concentrations used for cross-linking. Future work, though, is needed to see how this impacts gel mechanics. Tissue engineering for 2 decades has utilized these hydrogels to mimic natural tissue on a small scale, but continued advancements from results like ours push the world of artificial tissue engineering to new horizons. Thank you.
Hi guys, my name is Gayatri, and if you've ever been out in Arizona or California, then you've been exposed to a danger lurking in the desert. And I'm not talking the usual suspects, rattlesnakes, scorpions, bad drivers, or your mom, but a fungus known as coccidioides, which can cause a pneumonia known as valley fever. In serious cases, the spores of this fungus can travel into your bloodstream and your brain and spinal cord, resulting in a deathly complication known as disseminated coccidioidomycosis. Researchers found that the difference between you coming home unscathed after an encounter with this fungus or with a debilitating illness can lie in changing just one codon of a single protein known as STAT4. So I worked with my lab, the Genetically Engineered Mouse Models Core, in order to model this mutation in mice. We used a technology known as CRISPR-Cas9. It's relatively complicated, but I've decided I'd simplify it for you. In essence, it's like a giant pair of scissors, but scissors for your genetic code. It cuts your DNA at an exact location, so that when you introduce a new piece of DNA, it will glue itself or ligate itself into the old DNA. And if you inject this cocktail into, say, a mouse embryo, then voila, you get a mutated Franken mouse. And researchers can use our mice for a variety of purposes, such as testing treatments for valley fever and developing vaccines, so that next time you go out into the desert, you won't have to worry about a surprise sitting in the soil. Thank you.
Hello, my name is Vasu Kedia, and this summer I worked in Dr. Laksari's lab. I studied brain trauma in MMA fighters, and specifically I looked at kinematic data, such as angular velocity and linear acceleration from impacts to the head. Now this is important because there is a concern on repetitive impacts in contact sports and evidence of long-term neurodegenerative disorders. There is also limited research on the immediate effects of concussions in MMA fighters. Now for our experiment, the first step was to collect data. We had a mouthguard with a built-in gyroscope and an accelerometer, which registered data whenever a hit above 10 Gs was registered. We then had to filter the data and find the maximum peak for each impact. An impact is recorded over 104 milliseconds, and we only wanted to report the peak values. We then converted the linear acceleration of the mouthguard to the linear acceleration center of gravity of the mouthguard to the linear acceleration center of gravity of the head for more accurate results, and calculated brain strain. We calculated the peak max principal strain, which is the amount the brain stretches and deforms during an impact, and we calculated this for the entire brain, for the corpus callosum, and for the fiber strain of the corpus callosum. We chose the corpus callosum because it is a region commonly damaged in contact sports. Now we did this by using computational model. We had pre-trained neural networks which we entered the angular velocities in and outputted the brain strain. Now some results we found an average velocity of around 11.6 rads per second and an average linear acceleration of about 17.8 Gs. We also found a brain strain on the corpus callosum of about 0.103, while concussive impacts ranged from about 0.161 to 0.398. While most of our impacts were not within this concussive range, we did have some within the range. This is all important because understanding the responses of the brain will allow us to better predict and prevent these concussions. So then we also correlated the linear acceleration and angular velocity to the blood flow velocity. We saw that as the linear acceleration increased, the blood flow velocity decreased, which was unexpected, but we also saw as the angular velocity increased, blood flow velocity increased, which was expected. So this is all important because understanding the responses of the brain will allow us to better predict and prevent these concussions, and correlating the data such as the angular velocity and linear acceleration to the blood flow velocity immediately after the impact will give us a better understanding of the immediate responses to the brain in concussions. Thank you.
Hello, I'm Michelle, and today I'll be talking about my research on rescuing a mini-circle producing E. coli bacterial stock. So this summer I worked in the Van Dorselaer lab, and our lab works with the human papillomavirus, or HPV in short, which is the most common sexually transmitted pathogen and causes around 5% of all cancers. Now for my project, I worked with a special strain of E. coli bacteria that can be used to make HPV. So what's so special about this E. coli strain? Well, it has the ability to produce mini-circles, which are small recircularized forms of plasmids. In this case, our mini-circle is HPV. Now the problem is the strain of E. coli that we're working with already contains plasmids with HPV that we don't want. So our main objective was to cure or eliminate the plasmids in the bacteria using ethylene bromide. Now one unique aspect of our plasmids is that it has resistance to an antibiotic called kanamycin. So that means if the bacteria is no longer resistant to kanamycin, then it has successfully been cured. So after treating our E. coli with ethylene bromide, we spread it onto LB kanamycin plates, and we found that a few colonies grew on these plates. So while a majority of the plasmids had been cured, some weren't. So in order to find the cured colonies, we set out a second experiment. Here we picked 9 individual single colonies and patch plated them onto an LB agar plate and an LB kanamycin plate, and we found that none of the colonies grew on the LB kanamycin plate, which was a good sign because it meant that all 9 single colonies had been cured. So using one of our cured single colonies, we put it into a glycerol stock for long-term storage. Now, with our cured E. coli, the lab can use the bacteria to make other types of HPV to study how HPV causes cancer in human cells. Thank you.
My name is Keona Kuo, and this summer I worked in Dr. Nancy Horton's lab. And so our lab was actually one of the first to discover a process by which enzymes form chain-like structures, or filaments. And so glycolysis is a process that involves the breakdown of glucose to produce energy used to sustain cells. And my project this summer focused on the enzyme human phosphofructokinase 1 liver isoform, or HPFKL, which catalyzes a step of glycolysis. And so the question my lab is trying to answer is, under what conditions do filaments form? But before we could do this, we first had to purify enough protein to use in our future tests, and that is mainly what I focused on this summer. So to do this, we first infected insect cells to produce HPFKL. Then we used resin to separate out the protein from other unwanted cell matter. And finally, we put it through fast protein liquid chromatography, or FPLC, to even further purify it. After this, we ran a Western blot, as you can see on the left, with our purified protein, and there's this thick band at 88 kilodalton, which matches the weight of HPFKL, indicating that our purification was successful. On the right, this is the results of our FPLC run, and you see these 2 peaks. So we weren't sure which contained HPFKL, so we had to run an activity assay, and that showed that the first one contained HPFKL present to catalyze the reaction. Since we have now developed a protocol for purifying our protein, the Horton lab will continue to investigate the conditions under which filaments form and their effect on enzyme activity. And also, since HPFKL is necessary for glycolysis, knowledge of its regulation can help to inform new avenues for treatment of metabolic and other diseases. Thank you.
Hi, my name is Sneha Lakamsani, and this summer I worked in Dr. Gandhi Leroy's lab. My project dealt with autism and its diagnosis, specifically how boys are diagnosed 4 times as much as girls. Autism is a developmental disorder that affects 1 in nearly every 44 children in the United States. And there are many proposed explanations for why the gender disparity in diagnosis is so big, such as how estrogen gives girls a barrier from displaying common phenotypes associated with autism. So to see if some of these explanations are supported by the language used by health professionals, I screened through electronic health records from about 200 8-year-olds across the valley. Some of these kids were diagnosed with autism, others weren't, but all of them were screened using the DSM-4 diagnostic criteria. I looked at patterns and differences between who met what criteria out of the 12 options. In the end, I was able to find that girls showed higher frequencies of having speech development issues and conversational impairment, as shown by A2A and A2B on the graph. However, these differences occurred within the undiagnosed population, meaning that even though they showed these problems, girls remained undiagnosed with autism. After seeing such a stark difference between how boys and girls showed these symptoms in the undiagnosed population, I decided to dig a little deeper. So, to get a better idea of how diagnosing clinicians viewed these children, I looked at the top 6 frequently occurring adjectives in both populations. I was able to find that girls were more frequently described as being social, along with other more positively connotated adjectives like expressive, appropriate, and able. This aligns with usual explanations that girls are more adaptable and able to camouflage themselves socially in their environments compared to boys. For future studies, we can compare this data to the newer, currently used DSM-5 diagnostic criteria to see how diagnosis rates have changed over time. Also, we can use this to implement into AI autism diagnosis softwares to create more objective diagnosis to even out the gender disparity. Thank you.
Hi everyone, I'm Erin Lam, and this year I've been hosted by Dr. Yitzhak Zohar in his Organ on a Chip lab. So the first question we tend to get is, what is an organ on a chip? An organ on a chip is a plastic device that's been specially designed where we insert human cells in there, and ideally the cells grow and they replicate a true organ inside of a plastic device. And this has great implications because we can use this for things like studying cancer, because traditional models like cell culture dishes or a animal model get close to replicating the human physiology, but it's not quite there. So if we can have a model that closely replicates a human physiology with actual human cells, we could get amazing insights into how cancer works in an actual human organ without like the ethical issues of testing in an actual human. So basically, in a prostate, there are a few main cells, and currently there's been a model published that has two of the main cells, namely the stromal and the epithelial cells. And this has already proven excellent for cancer research, prostate cancer research, because those cells have shown how cancer develops in the prostate. But what we really like is a 3+ cell model. So here you can see the blue cells, which are the epithelial cells, and the stromal cells, which are the light pink cells on the bottom. But in the middle, I have added a smooth muscle cell. So now we're creating a 3-cell model to help study this. And you can see on the left, there are syringe pumps, which are pumping those cell-sustaining liquids. So here we can see we have these 3 different cells. They're separated by a yellow layer, and we're trying to grow them to replicate a prostate. So what we can see here is the microscope images I used to kind of show how this organ on a chip develops. So on the left side, we can see the smooth muscle cells, and they're kind of long and thin, and we can see they developed into a nice layer. In the middle, we can see BHPRS cells, which are another name for stromal cells. They're a little bit harder to see, but you can see on the arrows, they formed a nice layer. And on the right, we can see the IPEC cells, or the epithelial cells, and they formed a nice layer. So what we've shown in this is that a 3-cell model of a prostate is possible to develop, and this will help supercharge cancer research because we can get an excellent model for modeling prostate cancer, under-researched and fast-progressing disease.
Hello, my name is Emma Leon, and this past summer I interned in Dr. Schlenke's lab, which focuses on entomology. Throughout my internship, I continued a project that Dr. Schlenke has already begun, and it focuses on wasps, and more specifically on wasp eggs. Now, wasps are parasitoids of flies, which means that they lay their eggs into the flies, which will then hatch and eat their way out of the fly. However, a fly can only support a single wasp egg, so Dr. Schlenke wondered if the egg length and the amount of eggs a wasp carried affected its competitiveness. He found that eggs with a shorter length but that had more wasp eggs tended to have shorter hatching times. I looked at 7 new species and I dissected them in order to look at a single ovary under a microscope connected to a computer that used a program called CellScan Standard in order to view and count and measure the eggs. And in my graphs, I found that there was barely any significant difference between the wasps, and while I did find a negative correlation, it was very weak and it wasn't significant at p is less than 0.5. The lab does still hope to continue this project and watch the hatching times for these species in order to see if those factors affect it, and I would like to thank my lab, Keys, and the McNuggies for supporting me.
Hi, my name is Emma Leroy, and this summer I worked in Dr. Tyson Swetnam's lab. The purpose of my research was to improve the usability of community-gathered datasets for use with Google's Earth Engine. So the problem with working with these large geospatial datasets is that they often vary significantly in their organizational structure. This means that they can be difficult to use and often require significant amounts of pre-processing. One way to combat this is through the implementation of the Spatiotemporal Asset Catalog Specification, or STAC. This specification designates the creation of customizable metadata that then improves the indexability and discovery of these datasets. Google Earth Engine is a planetary-scale cyber GIS platform for geospatial analysis. Users can upload collections, which are groupings of datasets, and then write scripts to perform analyses and create visualizations of this data. For my overall project, I worked on Dr. Roy, a U of A campus colleague's Community Datasets Initiative. This is a GitHub repository of community-gathered datasets that Dr. Roy then curates for use with Google Earth Engine. There are currently 395 feature collections, which means that there's a significant variety in the structural organization of these data sets. Our overall objective was to align these data sets with the FAIR data principles. This means making the data findable, accessible, interoperable, and reproducible. To do this, we narrowed in on 49 of these data sets and worked to standardize the metadata using a template created by Dr. Roy. Initially, we went in and performed a preliminary analysis to assess the current organizational structure of these data sets' metadata. After this, we went in and edited 31 of these data sets to standardize them to the template specifications. This project upholds the principles of open science and thus promotes discovery, collaboration, and equity in computing. Thank you.
Hi, I'm Alex, and this summer I worked in Dr. Shrey's lab in the molecular biology department. Directed cell migration, a process known as chemotaxis, occurs in response to external chemical cues known as chemoattractants. Chemotaxis is responsible for many biological processes, such as basic wound healing. However, chemotaxis is also responsible for metastasis, which is the migration of cancer cells away from the primary tumor. In order to better understand metastasis, I researched how G proteins relate to GPCRs, especially within GPCR phosphorylation sites. Phosphorylation sites are essentially clumps of serine at the C-terminal of a GPCR, which are thought to affect G protein dissociation. Using site-directed mutagenesis and molecular cloning, I altered these serine sites into alanine or aspartic acid, which changed their effects on G proteins. I also added the enzymes R. luciferase and GFP for later use in BRED2 analysis. The model cell dictyostome was used, which can be seen in the video showing above, and that just shows chemotaxis within dictyostelium. The methods of my experiment included using PCR to essentially copy the DNA strands, inserting those strands into plasmids through digestion and ligation, then again multiplying those plasmids through bacterial transformation and mini-prep. After mini-prep, those plasmids were inserted into dicty through dictyostome transformation, which were then used in BRED2 analysis. In BRED2, a machine picked up the energy outputs of the enzymes mentioned earlier, which are luciferase and green fluorescent protein. A graph displaying these energy outputs is displayed, and when a decrease in energy output is shown, that means that the proteins are moving further apart from one another. With data such as this, we may guide the development of therapeutic strategies in preventing the spread of cancer cells. Thank you.
Hello everyone, my name is Renee Li, and this summer I worked in the biochemistry lab of Dr. Michael F. Brown researching the protein rhodopsin. Rhodopsin is a G protein coupled receptor, or GPCR, essential to light perception. GPCRs are important as they are targeted by 34% of all FDA-approved drugs. Upon activation by a photon, the retinylidene ligand, a molecule inside rhodopsin, isomerizes or undergoes a change in shape from 11 cis to all trans, inducing a conformational change in the wider protein. We used two computational methods to model two different aspects of rhodopsin activation. The first of these methods was molecular dynamics, which employed classical mechanics, or the physics you'd see in your everyday life. We also ran the computer program Gaussian to perform electronic structure calculations, which runs on quantum mechanical principles such as electron densities and wave function approximations. So, experimental data has previously suggested that upon activation, rhodopsin becomes more hydrated, with a difference of nearly 80 internal water molecules from the inactivated to activated state. However, a reanalysis of molecular dynamics simulations unexpectedly showed no significant difference in hydration between the two states. We also characterized the orbital structure of the rhodopsin active site with Gaussian. Using model systems of the rhodopsin active site, we were able to obtain a partially optimized model showing the qualitatively expected orbital structures settling on the retinylidene ligand. Understanding rhodopsin activation will allow for higher quality rhodopsin modeling. Additionally, as GPCRs are important in the pharmaceutical industry, understanding rhodopsin activation will allow for greater insight into GPCR function in general, resulting in the development of more specific, more effective drugs. Thank you.
Hi, my name is Sophie Li, and this summer I've been working in Dr. Jeffrey Pyong's lab in polymer chemistry. Polymers are long chains of monomers that can be used to make materials such as plastics. Specifically, the Pyong group focuses on the incorporation of elemental sulfur into polymerization, since sulfur is one cheap, and 2, a waste product of oil refining processes, which makes it a much more environmentally friendlier alternative. On my poster, there's a picture of a big pile of sulfur, which there are many just laying around in the world that could be used. This summer, we made sulfonyl chloride model compounds to better understand the polymer poly-S2-AMB-Cl2. Why model compounds? Model compounds are smaller and easier to understand while replicating the exact same properties as the polymer. Using techniques such as nuclear magnetic resonance spectroscopy and gel permeation chromatography, we looked at molecular structure, molecular weight, Abbe number, refractive index, and thermal properties. These properties can be illustrated in the graphs on my poster. We found good thermal properties such as high glass transition temperature and good thermal stability. We also found a high Abbe number and high refractive index, which are useful optical properties in imaging technologies. However, further research is needed to better understand its industrial uses. Thank you for coming to my TED Talk, and thank you to the Pyong Group for hosting me this summer.
Hi, my name is Byrdie Lopez, and I worked in Dr. Wei Wong's lab at the College of Pharmacy, focusing on chemical biology. When a cancer patient undergoes chemotherapy treatment, in some cases not all of the cancerous cells are killed off. Some of the remaining cells are induced into a senescent state as a result of the cancer treatment. Senescent cancers are larger, irregular in shape, but ultimately they activate new tumor growth and are especially difficult to kill with chemotherapy, making them a threat to the human body. My project aimed to determine whether the molecule ARV-771 is effective in killing senescent cancer cells. In previous research, it's been determined that ARV-771 is effective in killing cancer. However, it hasn't been determined whether it's effective in killing senescent cancers, which will be the aim of this study. The molecule works by binding together two proteins, a VHL and a target protein BRD4. When these two are linked together, it initiates the cell degradation process, killing the senescent cancer cell. Using Western blot and cell viability analysis, we were able to conclude that ARV-771 is in fact effective in killing senescent cancer cells. Moving forward, we wanted to look at the molecular interactions between the VHL and drug. By looking at these interactions, we can use them in future research to create a drug that only targets the senescent cancer cells. When used after chemotherapy treatment, a senescent targeting cancer treatment has the potential to eliminate all of the remaining cells in the cancer patient, giving them a longer life expectancy. Thank you.
Hello, my name is Zabdi Macias. This summer, I was hosted by Dr. Johnson's lab, which focuses on enhancing antibiotic treatment by better understanding how Streptococcus pneumoniae is affected by copper. Streptococcus pneumoniae is a disease-causing bacteria and the leading cause of bacterial meningitis and pneumonia in children and immunocompromised individuals worldwide. For 2 mutants and a wild type of Streptococcus pneumoniae, the relationship between the optical density and the cell density was obtained. Mutant Delta 727 does not accumulate copper due to genetic mutations. However, Delta 728 does accumulate copper. For these 2 mutants and a wild type, the optical density or absorbance was obtained by growing the bacteria in liquid media containing lactose or glucose, and then using a spectrophotometer to measure the amount of light that was absorbed by the solution. Afterwards, the cell density, or number of individual colonies formed, was obtained by diluting the solution and then transferring them to blood agar plates and then counting the number of individual colonies that grew to be visible. From this, the relationship between the optical density and the cell density was obtained. The results indicated that the 2 mutants reached a higher cell density at a lower optical density than the wild type. This may be because the mutants used up the sugar's energy more efficiently than the wild type. However, this mechanism would need to be more thoroughly researched. From these results, differences such as how the mutants grow and the size and shape of them can be determined through further research. This will allow more effective antibiotic treatments to be developed. Thank you.
Hi, my name is Jared Matheson, and this summer I worked in Dr. Eggers' research lab. Before I get into what I did, let me tell you about why this research matters. 1 in 29 Americans suffer from diabetic retinopathy. This is a complication from diabetes that leads to vision impairment, and in some individuals, complete vision loss. Now, these strange cells on the screen are microglia. They stretch throughout the retina and work to maintain a stable environment. They do this by communicating with other cells using neurotransmitters,
while also eliminating foreign particles and bacteria. This is all great within a healthy retina, but when blood glucose levels are elevated, such as in an individual with uncontrolled diabetes, these helpful cells become harmful. Rather than protecting the retina, they cause chronic inflammation, which contributes to the development of diabetic retinopathy. The lab I work in wanted to better understand how the structure and distribution of these cells changed as a result of these elevated blood glucose levels. So this summer, I analyzed images of mice fed a high-fat diet for 18 weeks and compared the microglia of those mice to mice fed a control diet. So what did we find? Well, to start off, we found a significant difference between the males and females in both the control and high-fat diet groups. This was surprising, and it's possible this difference was due to sex, but it's also possible that it's just correlated with the body weights. We also found a significant difference between the female control and female high-fat diet in terms of soma area, so the size of the cell body. This was expected because the cell body tends to grow larger as the blood glucose levels rise, but what was surprising is we didn't find the same difference between the male control and male high-fat diet. One possible reason for this was by week 18, when we did the study, the blood glucose levels, especially for the males, had started to converge. So if in the future we did this earlier in the study, when they were farther apart, we may find more significant differences.
Hello, my name is Joyeuse Mujambere, and I worked under Dr. George Sutphin in his lab researching the aging of C. elegans, or better known as worms. We use C. elegans to study stress and also stress response and aging in worms. So my research specifically focused on the process called hormesis, which is when you give an animal a sublethal dosage of a normally toxic physical or chemical agent, but in return it results in a beneficial effect for the animal. So our research found the optimal dosage and exposure timing to maximize lifespan by using fluorescence imaging and survival assays to determine lifespan increase and stress response. So how did we do this exactly? Age-synchronized wild-type C. elegans eggs were retrieved from the worms, then were allowed to grow to about age L4, which is the young adult stage. Then they were placed on agar plates containing various doses of arsenic. So we hypothesized that DAF16 and SKIN1 oxidative stress pathways played a key role in the life-extending factors of arsenic. To test this out, we gave the worms 50 to 100 micromoles of arsenic, and we saw that 3 out of the 4 genes that we tested showed that they were activated by arsenic's oxidative stress response oxidative stress. So my labs helped us understand how cells respond to stress simultaneously, and also how we can hopefully increase the lifespans of humans someday. Thank you.
So I'm Harlow Parkin, and this summer I worked in Dr. Shibley's condensed matter physics lab, where we look at the way matter behaves in 2D materials. Now 2D materials can be imagined as extremely thin sheets of some materials that tend to be around 1, 2, maybe a few atoms thick. So kind of like a piece of paper, except around 100,000 times thinner. So my research specifically looked at a device including chromium tridide, which is a ferromagnet, and indium selenide, which is a semiconducting material. Now both of these materials have a similar lattice shape and structure, but they both also degrade when they're exposed to light and oxygen, especially chromium tridide, as you can see from this top left image here. Now, to get around this and to be able to study these materials, we encapsulated them in hexagonal boron nitride, or HBN, which is just an insulating material. And to get these materials as thin as we wanted them, we used what's called the Scotch tape method, which allows us to place a bulk crystal on tape and fold that tape over on itself, splitting the material in half until it's about as thin as we want it. Then we transfer this new thin material onto chips, we look at it under a microscope, and we identify promising samples based on their color and their size. After that, we perform atomic force microscopy, which gives us a topographic image of the sample that allows us to determine the height and the cleanliness. After this, we actually start creating the device, which involves creating a stamp, which is a microscope slide with a little bit of gel and film that we put down onto the chips above the pieces, and we hope that they lift up when we lift up the stamp. And so we build the device from top to bottom. Now, unfortunately, this is where my research failed, although we were successfully able to pick up the top layer of HBN. The indium selenide and chromium tridide pickups did fail, so the device was not created. But why did we want to create this device in the first place? Well, in my lab, we use photons from lasers to excite electrons so they jump up to new bands, creating an exciton. But when you give electrons enough energy, they jump up to a new material, which creates interlayer excitons. And so what we hope to see here is a quantum exchange interaction where the chromium triiodide's ferromagnetism is transferred into the indium selenide. And this is cool because of something called the quantum Hall effect, which would give indium selenide superconducting qualities, allowing it to create a dissipationless circuit at room temperature, which is awesome because currently superconductors only work below their critical temperature, which is typically around negative 400 degrees Fahrenheit. And so creating a dissipationless circuit that can be used at room temperature would allow them to be placed in your everyday devices like your phone and computer, drastically improving their computing speed and power. Thank you very much.
Hi, my name is Corey Querimit, and this summer I work with the Churco lab to observe how they use mouse models and iPSCs, induced pluripotent stem cells, to study arrhythmogenic cardiomyopathy, or ACM. ACM is a cardiac disorder known for causing sudden death, especially in young individuals, and it's characterized by fibrofatty infiltration, cardiomyocyte death, and arrhythmia, which is irregular heart beating. Now, many mutations can cause ACM. However, a mutation in the PKP2 gene attributes to more than 40% of ACM cases. And with the increasing difficulties of using human models to study this disease, you had to default to using mouse models and iPSCs instead. And so what did I do? iPSCs are cells derived from blood or other types of somatic cells and have been reprogrammed using viral vectors back into stem cells, which can then be genetically edited to contain certain gene codes or mutations. And so what I did was I cultured my own set of iPSCs and differentiated them into heart cells, and then I compared them to a set that had the PKP2 mutation. I found that the mutated cells were a lot more flimsy and fragile compared to my own normal cells. And with the mouse models, I sectioned heart tissue from both our normal and mutated mouse models and stained them using various staining methods. I found that the mutated heart tissue contained less expression of the PKP2 protein, which is very integral to cell-to-cell adhesion of the heart's muscular tissue. Overall, the Churco lab plans to refine their methods of using mouse models and iPSCs so that we can continue to effectively and accurately study ACM. Thank you.
Hi everyone, my name is Yashas Raman, and this summer I worked in Dr. Gandhi Leroy's lab, which focuses on the gender discrepancy in autism spectrum disorder diagnosis. According to the CDC, approximately 1 out of every 44 children is diagnosed with autism spectrum disorder. However, boys are 4 times more likely to be diagnosed than girls. Autism can sometimes be difficult and tedious to manually detect. However, the existence of electronic health records and programming can help speed up the process. The algorithms employ a concept known as natural language processing, which is when computers try to analyze language in the way humans use it. I hypothesized that the most common nouns and adjectives used to describe children's behavior and development are used more frequently in boys than girls, which would imply that boys are more elaborately described than girls in these records. I analyzed the electronic health records of 200 children in Arizona using the Python coding language and NLTK libraries, which are used for natural language processing in Python. I collected data on the most frequent nouns and adjectives used to describe subjects and evaluated if there were any differences between boys and girls in the frequency with which these words were used. From my data, I found that there was no consistent trend suggesting that these words are used more frequently in boys than girls. In fact, most words were used slightly more to describe girls than boys. This data likely means that there is no inherent bias in electronic health records toward boys. If anything, they may be slightly biased towards girls, but I did not find conclusive evidence of this. Overall, my research finds that gender bias in electronic health records does not explain the gender discrepancy in autism diagnosis. Through deeper research in the future, such as more intricate analyses of electronic health records, certain instances of bias may be found, which may explain some parts of this discrepancy. However, nothing glaringly obvious shows a significant bias towards boys in electronic health records. Thank you.
My name is Mariam Rana, and I had the privilege of working in Dr. Andrews' lab this summer.
In Dr. Andrews' lab, I researched Ehlers-Danlos syndrome, which is a genetic condition that affects the connective tissue in the body by targeting the skin, joints, and blood vessels. There are 13 different subtypes of EDS, but specifically in my lab, I focus on hypermobile Ehlers-Danlos syndrome, also known as HEDS, and that is the only subtype of EDS where there is no known gene. Because of that, there is a lot of research in the world about this specific subtype right now. My lab specifically is a human research lab that focuses on improving the lives of those impacted by different genetic conditions and diseases and stuff like that. So our main goal was to see what we can do to help impact the lives of those affected by HEDS. So we had the question, what alternative treatments are HEDS patients using to assist with treatment management? So basically what I did was that my mentor and PI gave me interviews that have already been conducted and have been pre-transcribed, and I went through those interviews and I coded for all the treatment management and symptom management techniques that patients were using. So I went through and I created codes for every single one of them, and after I created those codes and gathered info on symptom management techniques, we put it into a software that allowed us to see the frequencies of similarities and differences in the way patients are treating their HEDS symptoms and what they're basically doing to manage the symptoms. And based on that, the results that we came up with was that 70% of patients reporting symptoms sought treatment, and this basically means that the decision to treat has to do with the quality of life rather than severity, and we kind of expected this because of different resources, time, money. Different people can treat different symptoms in different ways. And then just for reference, our main reported body systems were pain, immune system, and gastrointestinal. And basically what we're going to do with all this research is that the purpose is that it's going to provide alternative treatment for patients based on like peer reports. So basically we're going to see what works for one patient, and then it could potentially work to treat another patient symptoms. So overall the patients can help each other by us looking at the similarities and differences in the way that different treatment methods are being used. I want to thank my lab for giving me the opportunity to work in their lab this summer. It was an amazing experience, and thank you for Keys for this opportunity. Thank you.
Hi everyone, my name is Maritza Roberts Padilla, and this summer I worked at the Tefeli Lab of Ecosystem Genomics. Currently, monsoons have the potential to impact soil bacteria in semi-arid desert ecosystems like the Saguaro National Parks in the American Southwest. This is because monsoons will expose bacteria's networks of nutrients to variable change due to dry and wet oscillating conditions of rainwater. Furthermore, increasing amounts of greenhouse gases and the worsening of climate change have made it important for us to identify and understand the soil bacterial composition because they play a major role in the carbon cycle. This is because plants will fixate carbon dioxide from the atmosphere into the soil, and the bacteria in the soil will metabolize it and release it back into the atmosphere, creating a positive feedback loop. This is why my research focused on identifying and understanding the soil bacteria in the Saguaro National Park, where we took 4 different sample sites from this desert area, and after that we tried to identify the bacteria through a genomic analysis. And this was done through incubating bacteria, which we then ran a single colony PCR utilizing a 16S ribosomal DNA primer, which functions as a gene primer or a gene marker that amplifies a specific region and fragment of DNA for bacteria universally. After we did that, we sent 3 samples to sequence at the University of Arizona's core facility, and we got back results that told us that we had 3 different bacteria in 3 different genus, which were Streptomyces, Anthrobacter, and Massilia. Furthermore, we were able to phenotypically identify 25 different types of bacteria, which the Tefeli Lab will continue doing genomic analysis on and conduct a literature review. This information will help us create a more comprehensive view of the soil bacterial community in the Saguaro National Park and help scientists create more accurate climate change prediction models in the future.
Hi, I'm Akhil, and this summer I worked in the HAMMER lab. So my project involved measuring the expression of the SCN8A gene in human stem cells. SCN8A codes for a sodium channel called NAV1.6, and the specific mutation of SCN8A that I studied causes this channel to become overactive, resulting in neuronal hyperexcitability and epilepsy. The overall disease is called SCN8A encephalopathy. Now, moving on to the methods, the first step was to extract RNA from each sample. Then that RNA was converted into cDNA, or complementary DNA. And then finally, the amount of cDNA in each sample was measured using a process called qPCR, or quantitative polymerase chain reaction. So if we look at the graph on this slide, you can see multiple different curves, and each curve represents a single sample. And you can see that horizontal line, that's called the threshold line. The basic principle of qPCR is that the sooner a sample's curve crosses the threshold line, the higher the initial amount of DNA was in that sample. Now, using that concept and a bit of data analysis, these are my final results, and we can see that there are two major trends in this graph. So firstly, SCN8A expression increases as cellular maturity increases and differentiation progresses. So on the x-axis, I've listed the 4 cell types we use in this experiment from least to most differentiated, and we can see a general increasing pattern. Additionally, the second trend we can see is that compared to the control cells, the variant cells actually express slightly less SCN8A than the control cells. So overall, these results are important because understanding the mechanisms behind SCN8A expression could reveal potential therapeutic targets, as well as aid in drug development processes. Thank you.
Hi, my name is Samir, and this summer I interned at Dr. Dana Zhang's lab, studying a protein called NRF2. NRF2 controls a variety of your cell's protective mechanisms. However, an overactivity of these mechanisms can put these protections into overdrive, resulting in the cell being harder to kill and leading to diseases such as cancer. Because of this, in my project, I focused on trying to determine the structure of NRF2 when it binds to your DNA and transcribes and helps transcribe these protective genes in order to eventually be able to better design pharmaceuticals that can inhibit NRF2 and try to prevent this. To determine the structure, I used bacterial cells to make the protein, purified it by presence of a certain tag and size of the molecules to filter out all the other junk, and finally set up crystal trays that I periodically checked for crystals. In terms of purification, I was able to successfully purify my protein. You can see that in the top box, all the contaminant bars for the protein go away, while in the bottom box, the bars for our protein that we want are still there. With this, we were able to form crystals such as those seen in the first 2 images, which are microcrystals, meaning they're smaller and more rod-shaped, as well as clusters of crystals in the third image, which are larger and more fragmented. We were also able to make significant progress in determining the ideal conditions to grow these types of crystals in. Once we have a large enough single crystal, we plan on using X-ray crystallography for structural analysis, which involves shooting light at the crystal and studying the patterns that come out. With the structure of this complex known, scientists will be able to better design and develop pharmaceuticals that inhibit NRF2-based promotion of these target genes, which will eventually have major clinical applications for prevention and even treatment of NRF2-linked diseases such as cancer. Thank you so much for your time.
Hi, my name is Reeva Shrestha. This summer, I worked in the Chapman lab to learn more about heat shock proteins known as HSPs and to develop a selective inhibitor for a specific protein family, HSP70. Now, HSPs are molecular chaperones, meaning that they assist or chaperone in protein folding, and they also refold other misfolded proteins. Now, HSP70s in particular are normally expressed at low levels. However, when any stress factor is introduced, they'll become overexpressed. Overexpression causes them to lose their protein function, as well as inhibit p53, which is a vital tumor suppressor that normally functions to regulate cell growth. Now, the process to develop our inhibitor includes creating molecules and then testing them in various assays of HSP70 isoforms. I particularly focused on molecular synthesis. In this, I prepared reactions, analyzed molecules, and then finally went through multiple purification processes in order to isolate our desired reaction product. These processes include column chromatography, extraction, recrystallization, and finally filtration. One of the compounds that we created was compound ZXY0032. On the left here, we have the molecular synthesis pathway for this compound, also showing which other molecules were involved in the reaction. On the right here, we have our results. Isoforms A2 and A9 both have millipolarization levels below 0.5, indicating to us that our compound effectively binded to only these 2 isoforms. Now, this shows us that there were some aspects of our compound that were effective. And so in future experiments, we'll then pair it with another compound, bring them together as part of a coupling reaction to then produce a potentially better inhibitor. A successful inhibitor will play a significant role in preventing the progression of cancer, which will thereby increase the effectiveness of our cancer treatments. Thank you.
Good morning. I'm Nathaniel Vanderveen, and this summer I worked in the Space Treks lab under Dr. Jenkin Thanga to develop a semi-aquatic slug-based robot. Space has historically been a harsh environment because of cosmic radiation, as well as a lack of gravity, which both contribute to the degradation of the human body over time. However, terrestrial rovers have been used to prevent this degradation, but frequently break down in these harsh environments. Naturally inspired robots have used median paired fin, MPF locomotion, to undulate up and down on similarly harsh terrains here on Earth. I sought to develop SlugBot to use this MPF locomotion in order to take visual imagery of its environment. To do this, I used SolidWorks to model the individual components of SlugBot, and then I used C++ programming to test if the sensors and servos would work correctly. Overall, what I found was that if the servos oscillate in the counterclockwise direction to form a sine wave, SlugBot can move forward. However, if one of these sides or both of these sides moves in the clockwise direction to form a cosine wave, then SlugBot can move backwards or side to side. However, this means that in future iterations of SlugBot, that SlugBot shell needs to be segmented in order to avoid cracking and to shift with the constant movement of the robot. This also means that SlugBot on uneven and other harsh terrains such as sand can change the speed of oscillation of the servos, as well as the amplitude of the waves that are formed in order to traverse these other harsh environments. What this means is that in future space exploration and other missions, astronauts can be protected by deploying SlugBot. Thank you.
Hey everyone, my name is Andrew Way, and this summer I worked at the Gutraff Lab, which focuses on biomedical devices that are super thin, flexible, and implantable. And the way we protect these devices in the body is we use something called an encapsulation, which is basically a polymer layer that protects the device from water damage, chemical corrosion in the body, and mechanical strain. Now right now, the state-of-the-art method that we researchers use to test these devices for lifespan is we just dunk the device in a heated bath of phosphate buffered saline, or PBS solution. And what that does is it mimics the chemical properties of the body, but it doesn't capture the mechanical strain devices will face after being implanted. So here at the Gutraff Lab, we created Estira, which is a multimodal stretching stage, which basically just constantly stretches the device during the testing process. And that just ensures that mechanical strain is accounted for in testing encapsulations and the device itself. And that gives us researchers far more accurate lifespan estimates. So my research with Estira focused on the wireless data collection that it conducts on the device. Right now, if you're a researcher, you might only check in on your device being tested in the PBS solution every couple days, maybe even every couple weeks, to ensure that everything is running smoothly. However, with my research, I discovered that any encapsulation failure results in a significantly higher reported resistance value by our device in the testing stage. And that means that researchers know instantly how and when defects occur in the encapsulation, which really improves encapsulation research. Now, a great benefit of this is in the future, if these are consumer-grade electronics and I want to buy one as a consumer, I can be confident that if I'm exercising, playing sports, the device can withstand all that strain in my body. Thank you.
Hi, my name is McKenna Wheeler, and this summer I worked in Dr. Craig's Boundary Layer Stability and Transition Lab. So with any aircraft that you've flown on or maybe even just seen, including the ones from Davis-Monthan that rattle every building they fly by, there's wind tunnel testing behind that craft. Wind tunnels help us learn more about the aerodynamics of different objects, and hypersonic tunnels, like the one I worked on, focus on the aerodynamics of objects going Mach 5, which is also known as 5 times the speed of sound, or faster. Something that can be really hazardous in wind tunnels, though, is dust and other free-flowing particles. Aside from particle-based wind tunnel experiments, these particles can not only damage the equipment and the testing models, but in hypersonic tunnels, they even risk skewing the data, as they can affect the drag of differently shaped objects and create unnaturally occurring turbulence spots along testing models. To combat this problem, my project was to design and build a probe that would detect these particles inside the tunnel while it was running to better mimic what our testing models were experiencing particle-wise. The probe was made of 3 stainless steel parts with a Teflon adhesive attached to it so that the particles would embed themselves in the Teflon as they flew by. We were able to view with the microscope the craters that these particles created in the Teflon and saw 4 possible spots where this occurred. Unfortunately, our microscope was damaged, so we were only able to say we found 1 spot definitively, which we double-checked with a magnifying visor. But aside from our optical malfunctions, the probe was a success. This is now a tool that the LT5 team can use to make sure that their air is clean, which is going to be extremely important for them in the near future as they're getting a quiet nozzle installed in their tunnel, which is a very delicate piece of equipment that needs clean airflow. This is applicable too for other hypersonic tunnels, quiet nozzle or no quiet nozzle, to make sure that no data is in danger. Thank you.
Hi, my name is Veronica. I'm working in Dr. Xiong's plant and virology lab this summer. Our goal is to find a nanoparticle-based vaccine candidate. The pros of viral nanoparticle vaccines is that, first of all, it triggers immune response more effectively, and it's naturally adjuvant-free, which means that it benefits people that are vulnerable to the side effects of traditional vaccines. My specific research is testing the N-terminus of capsid protein of Pepino mosaic virus, shortened as PEP-B. The functionality of capsid protein is very crucial because it not only moves the virus from cell to cell, but also from a part of the plant to another part of the plant by hijacking the veins. I'm also testing how many amino acids can be removed from the N-terminus without affecting the functionality of capsid protein. By doing so, longer antigens can be displayed on the surface of the virus. However, because of the multiple functions of capsid protein, any change to it could potentially affect the virus's infection rate. Different mutants are being tested on 5 tobacco species and 4 tomato varieties. Delta 12 has 12 amino acids removed, Delta 25 has 25. PCR and mechanical inoculations are being used to detect the virus and analyze the functionality of the mutated viruses. Our results show that Delta 12 could basically maintain the same function just as the wild type, where Delta 25 could only infect 1 tobacco species and moves at a much slower rate. In conclusion, it is safe to remove 12 amino acids from the N-terminus of PEP and V, and that gives the antigens a larger space to be displayed. Thank you.
Hi everyone, my name is Shirley Xian, and this summer I've been working in the Translational Optical Imaging Lab with Dr. Dongkyung Khan and Ms. Momoko Sugimura. My lab is developing an in vivo confocal microscope for better eye imaging. In vivo confocal microscope basically takes a wave of light and sends it through a pinhole onto a point of the eye and constructs images of the eye point by point. Now this technology is good because it's non-invasive, it's rapid, and it's able to image various tissue depths. But the problem with it right now is because of how bulky and how expensive it is very inaccessible for a lot of people. So my lab is trying to create a more affordable and portable microscope for fast disease diagnosis. Now my research focuses on understanding how cornea movement affects the optical axis of the cornea. And this is important because if as the eye moves, the cornea's optical axis is changing, then how light travels through the eye and is refracted will change depending on the positioning of the eye, and this could lead to imaging error. So I'm studying this by taking videos of the eye and then running them through a MATLAB code. The code basically fits a curve to the cornea surface and then creates a circle from the curve, and then I track the changes in the radius of the circle and also the positioning of the center of the circle. This is basically tracking how the radius of the cornea is changing and also tracking the movement of the center of the cornea. And I found that the radius change was insignificant, and a lot of times it was due to things like your head moving slightly or your face twitching. And the radius I found through my code actually matches online data sources, so that's really good. I also found that cornea movement is significant, and it does have an impact on the optical axis, and this is because the cornea has a smaller radius compared to the eyeball, so it has to rotate more to match the rotation of the eye, so this leads to optical axis changing and imaging error. Now this understanding can help accommodate future technology to eye movement. Thank you.
Hi, my name is Brian Yong, and this summer I worked in Dr. Frank Duca's lab analyzing a potential mechanism through which dietary fibers improve metabolic homeostasis, or balance between energy intake and energy expenditure. Over 2/3 of Americans are affected by obesity, which is influenced by increased consumption of Western diets that are high in fat and low in fiber. High-fat diets impair the gut barrier, leading to increased lipopolysaccharide, or LPS, which is a component of pathogenic bacteria. This can then lead to the leaky gut and induce inflammation. However, my mentor Beth previously found that dietary fiber intake has beneficial effects on metabolic homeostasis. Fibers such as beta-glucan and oligofructose, when supplemented into high-fat diet, reduce body weight and adiposity. So with these findings, I hypothesized that dietary fibers will improve metabolic homeostasis by reducing inflammation in the small intestine and liver. During my project, I isolated RNA from mice small intestine and liver and synthesized complementary DNA by reverse transcription. I then performed RT-qPCR to measure gene expression of inflammatory markers called IL-6 and TNF-alpha. In my results, there was no significant difference in IL-6 and TNF-alpha mRNA expression in the small intestine and liver when compared to the control. Therefore, my hypothesis was not supported, as there was no significant change in inflammation upon dietary fiber supplementation into high-fat diet. In the future, the Duca lab will continue to investigate this mechanism further by analyzing inflammation in the adipose tissue and measuring circulating inflammatory markers. Thank you.
We would like to thank our University of Arizona faculty researchers, undergrad and grad students, postdocs, and lab staff who generously volunteered their time, talent, and treasure to mentor students year after year. We could not provide KEYS without your participation. Thank you to our KEYS donors for your support, giving KEYS the ability to provide a free and accessible opportunity for all interns to participate. This also allows us to continue engaging and mentoring our next generation of scientists for years to come. We would also like to thank our KEYS community for their support as well, including intern families, recommending teachers, our KEYS Advisory Board, the Bio5 Institute, and KEYS staff for their hard work and dedication that they provide to make KEYS a success each year. Congratulations, interns, for embracing your KEYS experience and working hard to gain new knowledge, new skills, and more genuine self-confidence. You joined the 576 alumni who came before you, and we know that you too will go on to accomplish great things. Your KEYS family will be here to support you in all of your future endeavors. We are so proud of all of you. Go and change the world.